This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 203, recorded on August 15th, 2019. I'm Vincent Racaniello, and you are listening to the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, hello there. How are you?
I just saw a picture of you on Facebook, smiling, you and Stan Malloy and a third person. Who was the third person?
His name is Woody Wood.
Woody Wood. He was an ASM president. 3 ASM presidents, yeah.
That's right. The three of us were ASM president, and somebody had the bright idea of having lunch together, so we did.
That's great.
Also joining us from Ann Arbor, Michigan, Michele Swanson.
Hello.
Did you see that picture on Facebook, Michele?
I did. Yeah, it made me smile.
They're all smiling. It was great.
What else can we do?
and from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
Charleston, you'd be smiling too because it's sunny.
It's sunny, but it's like a sauna out there today. We have 90 degrees, but it feels like 115 with the humidity.
Well, I commented on that picture that they must be smiling because of the San Diego weather, and Stan Malloy said, no, we're talking about microbiology. That's why we're smiling. And that's what we do here on TWiM. We talk about microbiology. We were having a conversation before on the origin of these words. I love to hear the origin of words. So first of all, you probably know this. I'm reading a great book by Erling Norby about history of Nobel Prizes. And there's a certain part where Salvador Luria was a faculty member in Bloomington.
That's right.
And Bertani was working with him. And you know who the graduate student was?
Jim Watson.
Jim Watson. And Watson.
Really?
And they were working with Shigella. They were trying to induce the phages from Shigella, right? And they said Watson was always freaking out because he didn't like pathogenic bacteria.
And Shigella, you only need 10 to get the trots.
So they had to develop a very rich broth to keep the phage from excising, to keep them lysogenic. Mm-hmm. And they called it lysogen broth, or LB.
That's right.
But I was told when I was a postdoc that it was Luria Bertani broth, which is wrong, right?
Well, it is, but it came out of the Luria Bertani lab.
Yeah, but they called it lysogeny broth because
that is indeed correct.
So I love learning things like that. Okay.
As a graduate student at IU, I heard that story from the woman. Her name was Helen Arthur. She was literally hired out of Bloomington High School, and she was effectively the media maker for the bacteriology department at the time. And so she is the one who helped Luria and Bertini and Watson assemble it. And she was still working in 1980 when I was a graduate student. And so Helen would tell the graduate student stories about Jim Watson as a graduate student and Luria wandering the halls looking for things and how she had to help the now 2 Nobel Prize winners get their work done. But Helen was a tremendous force of nature, high school educated, but she was a positively brilliant individual, and she really made the biology department hum at IU.
It's important not to forget these old stories. I like when people write them down.
Yeah.
If you like what we do here on TWiM, consider supporting us financially. Go to microbe.tv/contribute. All right. Even though it's August, we have some science for you. We're going to start out with a snippet from Michael.
All right. This paper is entitled Large-Scale Analysis of Human Microbiomes Reveal Thousands of Small Novel Genes. It's going to be published next week in Cell in the August 22 edition, and it was authored by Spiro, Freeman, Zelintny, Edfors, Grenfeld, Snyder, Pavlopoulos, Crepitides, and Bott, who are respectively in the departments of medicine, Department of Genetics at Stanford, one Codex in San Francisco, the Department of Energy's Joint Genome Institute, the Lawrence Berkeley National Laboratory, and the Institute for Fundamental Biomedical Research, Biomedical Sciences Research Center, Alexander Fleming, in Vari, Greece. And this paper hooked me with the following line: to support the transition of the microbiome field from descriptive science to a more mechanistic one, there's going to be an ongoing shift from 16S ribosomal RNA sequencing to whole metagenomic shotgun sequencing projects. But as my co-hosts are going to likely take me to task, that while we may be able to figure out who and what are present in a microbial community, developing this sense of wonder in the genetic diversity found associated with the niches of that our microbial passengers are harboring, without an ability to figure out the significance of their presence, if you will, linking the microbes or even their genes to a specific macro phenotype, like being too thin, too fat, developing metabolic syndrome, the transition to type 2 diabetes, that understanding is what's lagging behind. Yet we have companies and even clinicians out there encouraging people to consume prebiotics, probiotics, without sufficient evidence of their utility. So this paper is purely an in silico analysis paper. It's, again, part of the reason I selected to discuss it is it really is a call to action stations for the experimentalists out there. For the findings of these authors serve to open our eyes to this wondrous world that the microbiome, these remarkable communities and their small proteins, have to offer. They're going to help us appreciate that this symbiosis that we are an active participant in with our microbial companions. And we're going to learn that it's through their approach they found over 4,000 conserved small protein families from the human microbiome that will likely be fodder for many research projects to come. Just imagine, in the old days, it was you identify a protein, then you would ascribe a phenotype. That will generate 4,000 papers in the old days. So let's start by working through their graphical abstract.
I thought we started with the phenotype and then figured it out.
No, a lot of them were, the biochemists would isolate a protein, and that's what drove some of this merging of biochemistry with microbiology, and why a lot of microbiologists are great biochemists and vice versa. So I'm going to start by working through their graphical abstract to get a sense of what they did and what they learned. So we have a cartoon stick figure of a human, our source of the microbes, where on the image of the human, they actually they harvest 1,700 metagenomes, which our listeners will recall as the study of genetic material recovered from environmental samples. And in this case, the environments are from the human. And there are actually 4 of them that they actually harvested from. It was the mouth, the skin, the gut, and the vagina.
But Michael, if I could just emphasize the brilliance of this paper is this was all information that was already publicly available and shared in the science community.
Yes.
This group did not have to go and isolate 1,700 new genomes. Right?
That is precisely the point.
Yep.
That is precisely the point. In the next panel, you see their question or intent. And again, recall what Michele just said. This is their quest for small proteins, which they operationally define as being smaller than 50 amino acids in size. Now, I was a child of the first genomic revolution where you would sequence a gene and that would get you a paper. Today, it's merely a footnote in a paper. And, so there was a lot of work. And in our analysis, we would often discard the 150 bases that would give you a 50 amino acid protein because it was too small to be anything. And the authors comment that 90% of these small proteins that they refer to as small protein families have no known domain, which simply means there is no characterized protein that's present in the conserved domain database, or CC.
We don't know squat.
That's correct. And in the absence of the absence of this protein in CDD, where folks try to make sense out of the sequences, absence it's present in the database, there's not much known. So again, we don't know squat. So why is less than 50 or fewer amino acids important? Why are they absent from the CDD? Or why has this in silico community up to this point in time ignored them? Well, the answer to those questions are effectively their thought process in working through the database that Michele brought to our attention is what they did. They harvested the 1,700 genomes that were out there. But first, it's difficult to distinguish protein coding open reading frames from random in-frame genomic fragments, which result in their exclusion by the algorithms that make predictions that you do indeed have proteins. That they have traditionally been set to a higher threshold as being something that is real, or the open reading frame is significant. So that's one reason for an incomplete database. Secondly, in mutational studies, they were not likely the target simply because in the old days when you were doing knockout mutagenesis or random mutagenesis, you need a big target or a bigger protein because your likelihood of finding it, if it's bigger than 50 amino acids, increases, because if you're going to damage the protein, you're likely going to see it if you're not looking for a very small 150 base window that you have to make the deleterious mutation in. The other reason is biochemical approaches are not traditionally optimized for proteins this small. They're down in the weeds of the gel, if you will. And finally, experiments that rely on databases, such as those involving mass spec, often fail to identify these small proteins simply as a result of their absence. So now we understand the rationale of why they're hunting for less than 50 or less. It's because it's brand new territory, and the microbes make a lot of these small proteins, and they do indeed have many useful activity. So now we're still walking across the graphical abstract. In the next panel, they tell us that for each protein family or protein in their nomenclature, they'll offer us a taxonomic classification. The prevalence across the body sites, if they found it on the skin, the colon, the mouth, etc., and the type of protein that you're seeing, whether it's got a secreted domain, it's got a transmembrane domain, if it happens to have a predicted antimicrobial function. And then the next panel, they remind us of the locations from where the DNA was collected. And then using their computational pipeline approach, they identified and characterized thousands of these conserved and small open reading frames from the 4 body sites, leading to them to suggest to us that there are this diversity of unknown proteins and families with diverse functions from other animals, soils, that are found in the human microbiome, culminating in the last panel where they tell us what they have learned. And what they first learned is they found many of these small, less than 50 amino acid protein families, which are very abundant, are not assigned to a known protein domain, nor are they represented in reference sequences. And they found over 4,500 of these proteins. And when you look at their figures or walk across their figures, you see some of these proteins are found in as many as 753 different species of microbes associated with the microbiome samples that they collected from. And it's just absolutely fascinating tearing in to that first figure. You get the sense of how their pipeline or analysis rationale worked. And it's really quite remarkable watching how they discovered this. And they also then make a number of suppositions that I thought you might find interesting as you begin to consider the significance of their work. The other observation is proteins that play housekeeping genes or housekeeping roles for the microbes are expected to be unrelated to a specific niche or taxonomic clade. You know, if you're involved in protein synthesis, all bacteria make proteins, so you would expect there would be a lot of bacteria that would make these small RNA proteins that had not yet been characterized. But you scratch your head and you say to yourself, don't we know everything about protein synthesis? Well, that's where the bias comes in the sense that we learn most of what we know about protein synthesis from model organisms like E. coli. And E. coli happens not to express one of these small ribosomal RNA proteins that are prevalent across the tree of life of the microbes in this microbiome. And in fact, they identified 14 potential housekeeping families with 6 encoding different ribosomal proteins that were never known before. So they really are beginning to open things up. And why are ribosomal proteins significant? Well, we know a large number of our antimicrobials are against protein synthesis. Good example, good old tetracycline. It's attacking the ribosome. They also then continue, as they tell us what they've learned, they also address the arms race between the bacteria and the bacteria's nemesis for the millennia, the bacteriophage, which has led us to the evolution of the arsenal of anti-phage systems. We know them, of course, as restriction enzymes, CRISPR-Cas, and here they found 13 different protein families that they termed to be defense islands. And we all know about pathogenicity islands and resistomes, but here we now have a yet another term, a defense island, 5 of which encoded CRISPR genes, causing them to speculate that these small proteins are associated with already known or yet to be discovered defense systems. And that was effectively the subject of their 5th figure. And all the microbiologists who listen to TWiM often know about the opportunity that horizontal gene transfer offers microbes. And here they remind us of a 49 amino acid protein that has a membrane spanning domain that can enhance antibiotic efflux through the regulation of a multi-drug efflux pump. The one example that they use is ACRZ, and they identified 329 small protein families that they believe are horizontally transferred. One of the things I should point out is they similarly looked at a metatranscriptomic database. Once they got their 4,500 small proteins out, they asked the question, how many of these small proteins are indeed transcribed, which is the hallmark of whether or not something is real. And they found in the metatranscriptome database and the data they did in percent that indeed a number of these small protein families that they identified are indeed synthesized into messenger RNA, which leads us to believe they are actually made as indeed proteins. And finally, in their explanation of the 4 microbial niches, they learned that small proteins are potentially involved in crosstalk or inter-kingdom communication. Here they identified that over 25% of these small proteins that they've characterized, or 1,054 of them, are either transmembrane or secreted. And specifically, they called to our attention one of the families, 350 24, is very abundant across many mammalian guts and encodes for a predicted transmembrane protein that is encoded between a DNA binding protein. So you immediately think transcriptional regulation in an amidase gene that cleaves the cell wall. And in their discussion, they point out that a recent paper showed that a similar enzyme is involved in the formation of channels for material exchange between cells. And our listeners already appreciate that horizontal gene transfer between the bacteria and archaea occur. But the same is not true for the other domain of life, the eukarya. But using their taxonomic contig classification system, if you will, they're able to identify multiple families that map to more than one domain of life. And they then make the leap of faith that these proteins could facilitate transfer amongst the bacteria to the eukarya or vice versa. And finally, the highlight of this paper, which is why it's a call to action for the experimentalists out there, is they say to facilitate further investigation of these candidate novel small proteins, because they're only candidates until you have it in a test tube, is they provide a comprehensive resource file that is attached to the manuscript. It's the supplemental table 3 and also supplemental figure 7. And they collectively provide an exhaustive summary of the attributes associated with each of the 4,539 separate proteins that came out of their screen of these 4 microbiomes. And they encourage others to query the database of novel small open reading frames for families that obey specific attributes. They're actually charging the reader to go out and formulate a hypothesis as to what these small open reading frames may be doing. So in summing up, our knowledge up to this point in time, we know that we now are making large numbers of small proteins encoded by these human-associated bacteria. And before this paper, it was very limited. The data here and the computational approach that these authors offer us opens up a new frontier in the study of the microbiome and enhances our ability to exploit the therapeutic potential of these 4,500 plus small protein families of this previously ignored class of macromolecules. So it's really a remarkable piece of work. There's a lot more here, but I only had a snippet. And since it was in silico analysis, I really am looking forward to the 4,500 papers that will result from the characterization of all these proteins.
Good luck with that. You know, the NIH budget's not going up.
Well, you know, this speaks to the question that it should. I mean, Michele summed it up perfectly, and she said, these data are out there. They mined the data, and this is what they said. Okay, folks, here we have something new to think about. Yet I was talking to one of my colleagues last week, and her 7-year-old is, you know, not feeling well, and he's had an issue, and the pediatrician encouraged her to, you know, give him some probiotics to settle his stomach. And there's no data out there that said it's going to settle his stomach. So
what I was so impressed by with this paper is it really illustrates the kind of team science that we can do now. They point out in their acknowledgments that they have resources from the Department of Energy, from the Stanford Genetics Bioinformatics Service Center, the Proteomics and Mass Spec shared resource, and then of course they pulled all this genomic data from publicly available databases. They used algorithms to do their analysis and cut the data different ways. Again, all publicly available. And then, as you pointed out, Michael, at the end, they say, okay, we've generated this massive table of information. It is now publicly available. Have at it. Yeah. So I think this is a real tribute to how science works. And we do get great support from our federal agencies and from our home institutions.
It's a phenomenal piece of work. It's really exciting. And given the importance of the microbiome to the future of personalized medicine, I think a lot can be gleaned from studies like this. And for our undergraduate friends out there looking for projects for your students to begin to teach them about the genomic revolution and next-gen sequencing, this is a great paper to talk about with the methods, and I encourage Mark Martin to bring it to his micronauts and get him busy.
They also cite a couple of reviews from people who've been leaders in this field. So Gigi Stortz and also Pascal Cossart have published nice reviews on this kind of overlooked class of proteins, these small proteins, smaller than 50 amino acids in this case. So there is a lot of interest, and we now have the ability to go after this overlooked class of proteins. So I think it's super exciting.
Let me present another view, okay? Mm-hmm. I mean, this is great. I think it's wonderful, but 500,000 proteins, I don't even know where you start. I mean, don't we like science where you have a problem and then solve it? Whereas here you have 500,000 proteins, and what problem are you solving? So that's why it's a little puzzling to me. I think science is now moving towards this era of identifying lots of things, doing huge screens, but nobody does much follow-up because it's very hard. I mean, you can't study all these proteins. There's not enough people and money out there. So unless someone could figure out maybe that these participate in some process that they're interested in, then so we'll see. Let's see how long it takes for the first paper that takes advantage of it.
It gives you a different perspective or what the problems are. I mean, we thought naively that since we know so much about E. coli, we actually know a lot about E. coli. Well, it turns out it may not be true.
Yeah, that's fine.
We're missing one of its small ribosomal RNAs.
Yeah.
Right.
Well,
and I thought they did use some clever logic, saying, okay, if I'm interested just in microbes in the gut, let me not restrict myself to humans, but let's look at all the published gut microbes, whether they were in a worm or in a mammal or a small bird, and then if you make the assumption that some of the biology is going to be similar, but the genetic backgrounds are going to be quite different, then if you can pull out the subset that's shared across those very different organisms, it might lead you to some fundamental biology that we've overlooked. So I think there are ways to slice and dice it to narrow it down. So you're just using this as a sandbox, but not you're not studying every grain of sand.
I hope so, because I see so many screens where people pull out thousands of hits and then they never do anything with them. And they get these published in these high impact journals. And I would like to just do some investigative science again where you
absolutely
have problems. So I'm hoping that this will bear some fruit. But I just want to express my frustration with this. It's wonderful, but there's still some issues.
Like I said, this is a call to arms.
Yeah,
and you're making the call to arms, Vincent.
But this is not the first kind of screen that people have done. I mean, this is unique that they found these short open reading frames, but people do all kinds of other screens that are also call to arms. They put the data out there, and very rarely does most of it get followed up. It's a little frustrating, right? But I'm an old geezer, so don't listen to me. It's okay.
So do you think that we need to do a better job of educating ourselves and our trainees about all this publicly available data so that people can use it as a resource?
I think what we need to do is educate people on how to identify a problem and solve it, right? Because there are plenty of problems out there, but people like to take advantage of new technologies to do things like this. In this case, computational approaches. There are other screening approaches, but I think that we're not teaching students about the fundamental issues that remain that could be addressed by some of these approaches.
The designing of the binary experiment, getting the yes or no answer, and then iterating on the observation and moving forward to the next step as they work through the problem.
Yeah. I mean, there's a lot to be said for that. I mean, science is fundamentally changing because it's getting away from that and moving towards these large efforts. It's encouraged by NIH. They're putting together big groups that can do things like this, moving away from the investigator-initiated R01 type of project. I'm not convinced that it's the right way to go. If you look back in history, lots of great science done by small groups. Not convinced that big groups are any better. We'll see.
I completely agree. I think they're not mutually exclusive. Yeah. I would hope that the NIH recognizes that.
Yeah, I agree. There's a contribution for both, but
right.
I'm kind of seeing a movement away from smaller groups, and I don't think that shouldn't happen. I think there should still be. I was reading, as I said earlier, I'm reading a history of science, and they're talking about Andrei Lvov, right?
Mm-hmm.
He used to like to work with 3 people in his lab, him, his wife, and a technician. And look what he did.
Yep.
You can't do that anymore. You can't do it. It's just not possible to have a funded lab with 2 or 3 people. And I think that's a shame. Now, he didn't work on his own. He had other people around that he talked to all the time. But, big science is not the only answer. It's part of it, as Michele said, but it's not the only answer.
Well, and that's one of the things that you look for when you're going out looking for your first position is you want to join a department where you can find people that you can have unguarded conversations with that can help you think
totally
and help you explore. You don't want to move into a department so cutthroat where you keep your ideas so close to the vest for fear that your colleague down the hall is going to scoop you and, exclude you from, being able to do something positive.
All right.
Okay, let's go on now.
Thank you, Michael.
Yes. Well, I wanted a controversial paper, so we got one.
We have a paper suggested by our listener, Hannah, who said, I read this on Twitter and I think you would enjoy it. And Hannah, you are absolutely right. This was a pleasure. This is a paper published in Nature Microbiology and ectosymbiotic bacteria at the origin of magnetoreception in a marine protist. We have the first author, Caroline Montaille, and the last author is Christopher Lefebvre. And they come from a variety of institutions in Europe, including Aix-Marseille University, Sorbonne, Musée National d'Histoire Naturelle, Max Planck Institute in Potsdam, Germany, and an institution in New Zealand. Now, a long time ago on TWiM, we talked about magnetotacic bacteria. That was TWiM 25. Wow.
Long time ago.
Bacteria that have little magnetic particles in them so they can sense the geomagnetic field of the Earth and navigate that way. Magnetotacic bacteria. But they say at the very beginning of this paper, no study has ever proved that there are eukaryotes with magnetic organelles in them. They're called magnetosomes in the bacteria. So are there microbial eukaryotes, protists? So they went looking for them, and they went into the marine sediments off the coast of France. They went to a bunch of other places later, but initially the Mediterranean coast. They took buckets with them, and they sampled the sediment and a little bit of the water above it, and they brought it back to the lab. And then they do what they call magnetic enrichment. So they're looking for magnetotacic organisms.
And you can do this experiment at home. If you have a stir bar and some modeling clay, you can go get some sediment out of your stormwater retention pond, and you can put the magnet just above the sediment line, and you can literally witness the bacteria swimming out of the sludge, if you will, and accumulate like a colony at the pole of the magnet.
Yeah, that's what they do here. They use stir bars from the laboratory, big stir bars, which have two different poles, right? And they could put either pole against this. They have a tank with their samples in them, and they can see what's attracted to it. And they found lots of north-seeking bacteria, right? So they're attracted by the south pole of the magnet. But they also found some what they call atypical south-seeking organisms. Right, by using
There's
a
lot of diversity out there, even in microbes.
And so they took a look at these under the microscope. They're ovoid, they're striated, they're flagellated, and 16 microns long, 8 microns wide. They look like protists.
Mm-hmm.
So these are.
Guess what?
They are
protists.
They are protists.
It walks like a duck and quacks like a duck.
And so these are from the anoxic zone, where it's oxygen deprived, right? And so then they found this interesting observation when they put these protists on a microscope slide, and then they put a cover slip on top of them. They all broke apart, and voila, they see these 5 micron long rod-shaped curved cells coming off of the surface of these protists. And then so they did some electron microscopy, and it turns out that these organisms are really an assemblage. They are composed of bacteria, long bacteria that contain magnetic particles. And these bacteria are covering a unicellular flagellated protist, right? You have to kind of go look at the pictures, but you have this large protist, and it's covered all around by these thin bacteria. And in the bacteria are multiple magnetite particles. They say about 27 per bacterium. The bacteria are very long and thin. So they're arranged parallel to the motility axis of the eukaryote. And so the idea is that these bacteria are giving the eukaryote some magnetic sense. And they're stuck to the surface of the protist, they say probably by some mucilage. But the bacteria themselves, they align to a magnetic field, but they can't swim. They don't have a flagellum.
It's just absolutely incredible that these things are stuck on the surface, giving a sense of direction to the protist.
Yeah, the protist has the flagella, and they move, and the bacteria are steering in a way because they're magnetic and they're on top of this protist. Yeah, it's a nice little arrangement, right?
It gives a new meaning to the word backseat driver.
So this is a consortium, of course, or a holobiont is another word for it. So they say these are not really magnetotactic bacteria because the definition is that they are modal and these are not, but,
Is that picking a little bit?
Yeah, I think so.
Well, I don't know. Later they talk about the genomic reduction. So probably over the course of coevolution, they haven't needed their flagella. They use the host and save energy that way.
Yeah. So anyway, I think they have these particles in them. They align with magnetic poles. They're magnetotactic. But so then they went out again and looked in other places to see if they could find these in anoxic sediments. They go to different parts of the Mediterranean. They went to the San Francisco Bay. They went to New Zealand. Well, I guess that's where all the collaborators come from. And they found them in all these places, again, in the anoxic marine sediments. And it's interesting. The ones they get from the southern hemisphere have a swimming direction opposite.
I love that.
Isn't that great?
It's so beautiful.
It makes perfect sense.
And all of these look the same. They have these thin bacteria with magnetosomes. and they're covering a protist. The protist has two flagella that have been seen before. And they say the morphology of the protist looks like it's a member of the Symbiontida group, which are known to live in oxygen-deprived marine sediments. Okay, so that's...
There's a term they use, which I found totally amusing.
What's that?
It's the depth profile of oxygen fugacity.
Fugacity, yes. I looked it up, yeah, because I didn't.
I had to look up about 4 or 5 different words in this.
Yes.
When I was reading this paper.
On parle français ici.
Yes. So then they did some 18S and 16S ribosomal RNA sequencing, and they're taking the protist with its bacteria, right, and extracting the RNA. And that sequence showed that, in fact, the protist is a member of the Symbiontida with a very close species called Calcincia aureus. And the bacteria seem to be related to the delta proteobacteria, which are known to be sulfate-reducing bacteria. And they show that the phylogenetic tree of these sequences suggests that these two partners, so this is a symbiosis, or we think it's a symbiosis, they co-evolved from a single ancestral symbiosis. And if you look at the evolution of the sequences of the protist and the bacteria, the delta proteobacteria, they looks like they both started together a long time ago and have evolved together. All right, so the topology of the trees are what we call congruent, so that they co-evolved and diversified.
I thought that was also really beautiful. I'm sure that just gave them confidence that they really understood what was happening.
Yeah.
These different approaches are all pointing to a similar conclusion.
So this is a symbiosis. We have multiple different organisms living together. So is it a mutualism? What's the fitness game for each one, right? So they decide to sequence the genome of this bacterium, and the whole genome, they have 98% complete sequence. It's 3.2 million bases. It's a little genome there. And they see that there's lots of genes that are not present. For example, there's no genes encoding flagella or flagellar proteins. There's no genes encoding parts of chemotaxis pathways. These bacteria don't have to go anywhere. They're stuck on the protist, and the protist does all the driving, right?
Does all the moving.
Does all the moving. And they move according to the magnetic field, which is dictated by the bacteria. This is great. I really like it. So then they find what they call a magnetosome gene cluster, which is typical of magnetotacic bacteria. And so they say, well, the bacteria give magnetic sensing and the protist gives motility, but there's probably more than that. And so then they begin to speculate a little more. So what's the benefit for the protist? What's the benefit for the bacteria? I mean, you can see that beyond the magnetic attraction, what's going on here. Well, the protist, of course, could navigate towards chemicals and nutrients that they both could take, for example, and they speculate that the shell formed by the bacteria might be protective.
Would you say that the bacteria, the GPS for the protist?
Yeah, that's a good way to look at it, yeah.
But how do they send the instructions to the protist to tell it to go up or down?
I think the protists just swim, and then the magnetic field keeps it in line, right? It's like a track.
Okay.
It's just the flagella are running, and if there's a magnetic field, that's it.
It's going to be locked in because of the sheer mass of the number of magnets present by the number of protists attached to the shell of the.
Right. That's the point, really.
Yeah.
Yeah, exactly. So then they say, so there's got to be some metabolic trade-off, right? So let's look at the gene content. And so if you look at these bacteria, the genes in the bacteria, it looks like they're strict anaerobes, and they do sulfate reduction and probably oxidize some compounds like other members of this desulfobacteriaceae do. And so they have a number of genes that they identify. They identify genes involved in sulfate reduction, hydrogen oxidation.
Well, that's what the bacterium is eating via the
Yeah, this is the bacterium.
The bacterium's eating the hydrogen.
The
and it's reducing the sulfur,
right?
The sulfate. So are they feeding the protists the H2S, or is that just going off as gas?
And then they have a pathway for carbon dioxide fixation. So they say these bacteria are probably growing with hydrogen as the electron donor, sulfate as the electron acceptor, and a carbon source of carbon dioxide. And so then they look at the protist. The protist has in it a layer of organelles. It looks like mitochondria, which they say look like hydrogenosomes. They make hydrogen. And so they're thinking that the protist makes hydrogen, which is then used by the bacteria. So they're acting as hydrogen sinks, right? And again, they found genes involved in hydrogen oxidation, which would make sense. And then the protist is making acetate, molecular hydrogen, and carbon dioxide, and this could get into the bacteria, which in turn could use it. So the protist has a hydrogen sink, so the hydrogen gets taken away. And they say they probably also take advantage of bacterial primary production of organics, maybe by diffusion, although they don't have any evidence, but they say maybe the protists actually eat the bacteria.
Well, no, they may be giving them the glucose because
they say they're eating it. They say they're eating it.
Telltale signs that they munched on some.
They munch on them.
And they might be cultivating these bacteria as a kitchen garden.
Yeah, several observations indicate that these protists are bacteriovorous phagotrophs capable of consuming their ectosymbiotic bacteria. They eat the hand that feeds them.
But not too fast. But not too fast.
But not too fast. And they maintain the number of magnets on them because it's a kitchen garden. They don't want to trim too much of that basil.
Yeah. So they let it grow. They eat a little bit, and they
I was also wondering if that might have been some of the early selective pressure for these microbes to stick on the outside of the protist so they don't get swept into the mouth.
That's an interesting idea.
Oh, that makes perfect sense.
Oh, my goodness.
And then selected for that. And,
Cytostome.
Kitchen garden, yeah.
So as the bacteria divide, they push the one off the edge towards the mouth?
Maybe. Yeah, there is a little mouth there, yep. The feeding apparatus, right?
Yeah.
Right.
Yeah, that's right. They actually use the word kitchen garden, yep.
Mm-hmm.
So they say we need to grow these in the laboratory and do some more work on them to sort this out. But they say, in concluding, this discovery, it's a magnetotacic symbiosis, challenges our vision of the diversity of magnetically sensitive organisms. And this, of course, is the first discovery of a magnetic eukaryote, microbial eukaryote. And it's a symbiosis. So we still don't have any eukaryotes with magnets in them. No unicellular eukaryote is currently known to produce magnetosomes by itself. It's always, as far as we know, it's a symbiosis where the bacteria have the magnets.
And this may happen with other organisms, right?
It might, yeah. They actually talk about some algae where there might be similar things happening, microalgae. Anyway, so that's the story. I think it was just lovely.
Really cool.
Start with going to the marine sediment and pulling out some sediment, and here you have these fascinating things which you've never seen before and give you a lot of insight into evolution and magneto sensing.
The way they described it, too, is they had their protist, the large one, and they were putting it on a cover glass with a cover slip on top. And then probably they just saw all these tiny rods just flying away from the protist. And that's what led to this understanding that it was actually a community.
Yeah. As soon as they put the cover slip, they say they broke apart and they saw these little things coming off, which they hadn't, they didn't know they were separate. They thought it was part of the protist, right?
Yeah, and because it's August, I wasn't able to get in touch with the authors. I think they are on a well-deserved holiday. They are down in the Mediterranean. But I would have loved to have heard the story of the person who did that first experiment, put it between a cover glass, and then saw those bugs flying off the protists.
Yes, for sure.
A eureka day.
Yep. So anyway, Hannah, you are right. We did enjoy it very much.
Yes.
So thanks for sending that. All right, let's do a couple of email here before we finish up. The first one is from Ryan, who says, Dear TWiM, we have a case study to look at 5 LA Police Department officers with typhoid fever. And he sends us a bunch of links to this, and there are a bunch of stories. If you Google it, you can find there have been in the past few months cases of typhoid fever in police officers in Los Angeles. And I've looked around, and we really
if they don't get shot, they get typhoid.
Well, the question is the police department going to recommend that they get the typhoid vaccine? Because there's a nice attenuated vaccine out there. You just drink it, and you'll get immune to typhoid fever.
I guess they could make it available to them, right?
Yeah.
You can decide. I mean, this is a foodborne, waterborne illness, right?
Yeah, absolutely.
And so they don't know what the source is. It would be nice if they figured it out. Do a little, what, as Michael calls it, leather shoe epidemiology.
Shoe leather epidemiology.
Yeah. According to the Newsweek article, about 350 people in our country are diagnosed with typhoid fever, and typically they're travelers. They've visited places where typhoid is endemic, like India, Bangladesh, Pakistan.
Well, it would be easy to find out from the police officers if they've traveled or not, right?
Or they encountered individuals that have recently emigrated from those areas.
Yeah. And Anthony writes, greatest invention in the prevention of infection, the widespread use of cotton clothing that allowed for aggressive washing, including boiling, has been considered a major advance in health. This allowed for the control of arbovectors. Cleaning wool is tedious. Before cotton made a clean change of clothes convenient, people tended not to wash themselves or their garments. This is from a book called The Biology of Human Longevity by Caleb Finch. How about that? Cotton.
Hey.
Amir writes, Dear TWiM team, hello from Jena, Germany, where it's cold but sunny. My name is Amir, and I actually wrote to you a while ago from sunny Israel and was really pleased with the answer you gave me, so I decided to write again. I'm doing my PhD on fast diagnostic methods for clinical labs. My project is about fast detection of resistant strains, but I was wondering what kind of test or faster, cheaper test is needed today in the clinical labs. I asked different people this question, got a lot of different answers. For example, a way to evaluate microbial load in drinking water, the same but only for specific pathogens like Legionella or some algae, a fast way to determine which antibiotics will work, fast way to identify which specific bacteria is in a urine sample. What do you think? What is missing today? Also, thanks a lot for the hard work. I'm really enjoying the podcast. So I sent this question to our friend Robin Patel.
Our president.
And our leader, president of ASM.
President of ASM. She said, I will respond from the standpoint of clinical, medical human microbiology laboratories. The water questions would best be directed to a public health laboratorian. I agree that we need fast strategies to get patients who need antibiotics on the best antibiotics for their particular infection, to avoid overtreatment of those with bacterial infections who need antibiotics, and to avoid giving antibiotics to those who don't need them. I think the last point about urine is in line with this, but whatever test is developed and deployed needs to be actionable, and that action needs to make a difference in some way, ideally to address the points outlined in the prior sentence. Hope this is helpful. All right. Thank you, Robin. And one more. Anthony sends us, this is very interesting, an article in the BBC and also a link to a journal article. Progress in the development of vaccines against rumen methanogens. The idea is reducing methane emissions from cattle by vaccinating them.
So you'd vaccinate against the methanogenic bacteria, the archaea, in their stomachs.
Yeah. Don't they need them?
I find it difficult to imagine how, even if a cow is making a lot of antibodies, the antibody would find its way into the rumen, which is where the methanogens are.
Yeah, the concentration high enough to deplete them.
Yeah. So the idea here is, of course, to reduce methane emissions. And some people are suggesting we don't eat as much meat, of course. So who was this? Pat Brown, right? The Impossible Burger. And Pat spoke at ASM Microbe, right?
Yes. Yep.
And he wants to make non-cow-derived burgers, which is one way. And the other way is to vaccinate cows. So this is very interesting.
So the way this story ended, he asked a question, I wonder how much methane herbivores dinosaurs released. And so, Elio, this could be a good Talmudic question for you. How much methane did the dinosaurs release? And how would you do that calculation?
I'll use that.
That's an interesting. There were tons of them, too.
Oh, yes. Yeah. There were millions, billions maybe, of dinosaurs.
But they didn't have exactly a rumen. On the other hand, non-ruminants also make methane, like horses.
Yeah.
So, there were herbivores. Yeah. There were a large number of herbivores. So it's a good Talmudic question.
Yes, sir.
How much? Well, you could start with a cow and figure out how much they release per gram of weight, and then scale it up, right?
Yes.
All right. That is TWiM 203. You can find it on your favorite podcast player, and if you do listen on a podcast player, please subscribe. You can do that by pushing the button that says subscribe, and you get every episode automatically. And we know how many of you are listening. We'd like to know that. It really helps us a lot. And if you like what we do, consider supporting us. You can go over to microbe.tv/contribute. We have different ways that you could give us, say, $1 a month or more if you feel so driven at Patreon or PayPal and so forth. It would really help us with our expenses. Like traveling to see Elio once a year in San Diego, right? That would be a good expense.
Yeah.
Would you like us to come once a year, Elio?
Yeah, would I love that? We can talk about some other subject then.
Yeah, we can. We can.
And finally, we love getting your questions and comments. Please send them to twim@microbe.tv. TWiM, speaking of TWiM is Michele Swanson. She's from the University of Michigan. Thank you, Michele.
Thank you, and I wish you all a happy end of August.
It's the last 2 weeks.
I know.
It's so sad. It goes so quickly, doesn't it?
Oh, gosh.
Oh, well. Well, next week I'm going to Sweden for a week, so.
Okay,
nice.
To do a couple of 3 TWiV podcasts. I'm going to do that.
Wow. The sun never sets, though. You're in eternal summer over there.
Is that right?
I don't think the sun goes down until like 11:30, 12:30 at night.
It's funny, when I was arranging for all the guests on my podcast there, a lot of them said, well, this is August. You might not find people in Stockholm anymore. But I did. It's so funny. Elio Schachter's at Small Things Considered. Thanks, Elio.
My pleasure. Thank you so much.
Michael Schmidt, Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
Vincent Racaniello. You can find me at virology.ws. I want to thank ASM for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.